Thank you, everyone, and I'm sorry we have to start the day with such sad news, but we're going to move ahead with our science talks today. The first talk will be called Immersive Genetics 101. You guys have all already kind of been immersed in the world of genetics, like it or not, but we're delighted to include Tess Levy, who's the Assistant Professor of Psychiatry and Clinical Trials Manager at Seaver Autism Center in New York at Mount Sinai University. She is an expert in genetic counseling. Many of you have talked to her or worked with her before. She is going to show us she's going to explain the genetics. The genetics is changing every day, right? So, of course, the gene is the same, but what we know about other genes, what we know about the mechanism, what we know about different types of mutations is an ever-evolving thing. So she's not only going to explain that, but she's also going to talk about how to read a clinical genetics report, which is something I know you guys have been dropped in the deep end of the water on. She'll guide through interpreting those reports. She'll cover different types of genetic testing, what you should know about follow-up procedures, and how specific genotypes may correlate to symptoms or features that you're seeing in your kids. She has really been invested in helping PMS families for a while now. And you are very, very lucky. I've heard her give talks before. She does this in a very clear, understandable way. So you are going to be very, very relieved about not being overwhelmed. So thank you, Tess.
Hi, everyone. I am Tess, as Alicia just said. I work at the Seaver Autism Center in New York, and I have spent the last about 5 years looking at Phelan-McDermid syndrome and specifically at the different genetic causes of PMS. Okay, thank you. And I think also over the last 5 years, I have been lucky enough to meet now over 100 families who have kids and adults with PMS, and just feel very lucky to be part of this community. So today, I have been asked to cover a few different topics. The first one is going to be Genetics 101. So we're going to start all the way at the beginning and talk about what a chromosome is, what a gene is, what a mutation is versus what a deletion is. And then we will go into the types of genetic testing that may have diagnosed your children or your loved ones, what that testing can see and what it cannot see, and then we'll look at a few examples together of how to read a report. I asked also the foundation to let you know that you can bring your reports here, either in paper or on your phones, and when we get to this part, I welcome you to open them up, and then you can follow along and hopefully have a better understanding of your child's report after this session than you did coming into it. The last thing that we will talk about is some follow-up testing. So once your child has a diagnosis of PMS, what do you do next? What genetic testing do we recommend? Why do we recommend it? And what can it tell you? The first thing I just wanted to get a sense of is how comfortable everyone feels with genetics. So if you guys can show me with your hands, 1 being I don't feel comfortable at all, to 5 being I feel pretty confident in genetics, or at least confident in the genetics of PMS, with a 2, 3, 4 being in the middle. Nice. Okay, perfect. And then the second question, just to get a sense of how much time to spend on each concept, if you can show me a 1 if you know your child or loved one has a deletion, and a 5 if your child has a mutation, with a 3 being you're not quite sure, and hopefully the 3s, again, by the end, will have a better idea. Okay, perfect, thank you. I kind of expected a mix of 1 to 5, so we're gonna start all the way at the beginning, and then get to some more advanced concepts later on in the talk. If you feel very comfortable in genetics, feel free to tune out at the beginning, and if you don't, feel free to tune out at the end. It's really listen to anything that you would like. So starting with Genetics 101, I'm going to show you an overview, which you might have seen before, and then we're going to start back at the beginning and look at things in detail pertaining to Phelan-McDermid syndrome and kind of zoom in on each genetic component until we can't zoom in anymore. So this diagram, you've probably seen things like this before, but it shows you that our body is made up of millions and millions of cells. If we take a cell and we look inside of it, we see all of our genetic information, which is stored in chromosomes. If we take a chromosome and we look within that, we see a long strand of DNA. Shorter portions of this DNA are genes. Typically, our body reads through one gene and it makes one protein. That protein goes off in our body and does its job, whether it's things like determining our hair color, our eye color, but also how we develop, how our brain functions, how our brain cells talk to each other. This is the basis of all genetics. So what we'll do now is start at the beginning and then look at how PMS fits into this. This is the result of a karyotype, which is a type of genetic testing that we will look at a little bit later. But basically what this does is it takes a photo of our chromosomes and then it lines them up from biggest to smallest. So you can see that we have 23 pairs of chromosomes. They're labeled 1 to 22. And then we have sex chromosomes. This is a karyotype from a male because you see an X and a Y, whereas a female karyotype would have 2 X's. So you see here that we have 2 of each chromosome. We get 1 from our mom and 1 from our dad of each chromosome. Within these chromosomes, we have genes. So again, we have 2 copies of each gene, 1 on each pair of the chromosome. The chromosome that we know that's important for Phelan-McDermid syndrome is chromosome 22, which is the smallest chromosome all the way at the end of this karyotype. So if we zoom in just on chromosome 22, we can see the organization and structure of this chromosome. At the top, we see the short arm, which is also called the P arm, and at the bottom, we see the Q arm, which is also the long arm. And then within here, we can see the banding or how it's labeled. So you can see on the Q arm, it starts at 1.1, it goes down to 1.2, and so on. So when you see 22q, you know that that's chromosome 22, and then it's the long arm or the Q arm. And the region that's important for Phelan-McDermid syndrome is at the band 1,3. So that's what 22q13 stands for. And the region even more specifically that's important for PMS is the region 13.33, which is all the way at the end of this chromosome. And if we zoom in just again at the end of this chromosome, we can see all of the genes that are within this region. So this is just a cutout of the end of the chromosome, and you can see that there's many, many genes in this area. This is just a few of them. And all the way at the end, you see SHANK3, which is a gene that's colored in red. This is the gene that we know that's responsible for causing Phelan-McDermid syndrome. And you can see it's all the way at the end. It's actually the third to last gene that's on chromosome 22. And if we take SHANK3 and we zoom in just on that, we can see the structure of the gene. And if we zoom in one last time, we can see that the gene is spelled out with individual bases or individual letters, which spell out all of our genes. So you can see here underneath the gene, the letters A, C, G, and T, which are the 4 letters that spell out all of our genetic information. This is the most that we can zoom in. So we've gone all the way from a chromosome to a gene and now to the letters of the gene. One last concept, which I'll introduce now, is just how our body can read through genes. So our body reads through 3 letters at a time. It kind of jumps 3 letters, 3 letters, 3 letters. And it starts with 3 letters that tell the body the gene starts here, and then it continues to jump down. And then at the end, there's 3 letters that tell our body the gene ends here. This will become important later when we talk about how genes can be spelled incorrectly or how there can be mutations. For people who are less genetically inclined, there's a pretty well-known and well-used analogy that I'll introduce. So you can think of all of our chromosomes as like a library which holds and stores all of the books in the world. You can think of one chromosome as like one bookshelf which stores and organizes a set of books. You can think of one gene as one book which holds one story, one recipe, one instruction manual. And then you can think of bases of a gene as like letters in a book that spell out each recipe or each story. I will keep this analogy kind of at the bottom, moving through the rest of the slides, just in case you get a little lost. I hope that this will reorient you. Moving on to the types of genetic variation that we see in Phelan-McDermid syndrome. So we know that there are two main causes of PMS. One is at the level of a chromosome or a deletion. So there are some kids who have 2 copies of chromosome 22, but the end of one of their chromosomes has been deleted. For other individuals, they have both copies of chromosome 22, but one of the SHANK3 genes within the chromosome is not spelled correctly, making it not functional. These are the 2 overall types of genetic findings that we see, and I will explain in more detail, starting with deletions and then moving on to mutations. So again, deletions at the end of chromosome 22 cause PMS. Up top, you can see a full-size chromosome, and then a chromosome which has a deletion right at the end. If we think of this in the library, we know that we can have a bookshelf, and then this other bookshelf is missing 1 row of genes. So it's missing a row of stories or a row of recipes. We also know that the size of the deletion and also therefore the number of genes deleted are different between people with Phelan-McDermid syndrome. These are 3 different examples. The first one represents the biggest deletion, and you can see how much of the chromosome has been deleted, which then corresponds to over a row of books. There in the middle, there's a middle-sized deletion, which cuts off a little bit less of the chromosome, and also therefore only 2 sections of the books are missing. And then in the last example, you can see a very, very small deletion where there's only a handful of books that are missing from the bookshelf. Overall, we know that there are many sizes of deletions that cause PMS. The one thing that ties everyone together is that the SHANK3 gene or the SHANK3 book is always deleted for everyone with PMS. The other concept I wanted to bring in was a ring chromosome. So it's about 10% of kids who have a deletion also have a ring chromosome, and this is when, in addition to the bottom of chromosome 22 that is broken off, the top is also deleted, which causes the chromosome to form into a ring. I didn't have the best library analogy for this one, so I didn't add it, but I hope this at least can help you visualize how a ring chromosome is caused. The other type of genetic variation that we see in Phelan-McDermid syndrome are mutations. So again, these are kids who have both copies of chromosome 22. There are no deletions there, but one of their SHANK3 genes is not spelled properly. So these are changes that are at the level of the gene or level of the different letters or the bases. So in this example, you can see that the letter C, the second letter, was changed to the letter T, which seems like a very small change, and it is. But sometimes these small changes can really affect the meaning of a sentence. So in this example, you can see the sentence that started as the cat ate the rat changed to the hat ate the rat, which suddenly makes a lot less sense. And just like we see different sizes of deletions in PMS, the location and the type and the specific change of mutation that we see also is different across different people with PMS. So again, these are 3 separate examples of different types or different changes that we can see in SHANK3. So the first one that we see is the letter T was deleted, so just 1 letter is missing. In the second example, we can see that the letter G was duplicated, so now there's 2 G's instead of 1. And in the last example, we see that the letter G was changed to the letter A. So what these changes are, in the end, the specific change doesn't really matter, but we know that all of the changes or the mutations we see in SHANK3 cause the gene to end early, which is also called premature termination. And when we think of this in the reference of our analogy, we can think that when we start a book and we're reading it, when we reach the sentence that doesn't make sense, instead of continuing to read the book, we just close it and put it away. It doesn't make sense, so we're not going to continue reading it. And one of the reasons for this goes back to how our body reads through genes. And so we were talking before about how we read 3 letters at a time. And so when we affect just 1 base or just 2 bases, we know that in addition to changing that 3-letter sequence, everything after that is also shifted. So once we delete 1 base, it's going to change that meaning, but everything after that will be different. And so this is called a frameshift mutation and one of the most common types of mutations we see in Phelan-McDermid syndrome. So this was overall genetics. I hope you have an understanding about genes and chromosomes and the different types of variation that we see in PMS. Now I will go over the most common types of genetic testing that you all may have had, and then quickly go into how to read your own reports. So starting again with deletions, and then we'll move on to mutations. The most common test that we see that picks up deletions is called a microarray. What this test can see is any changes in the amount of genetic information somebody has. So it can see deletions or missing information, it can see duplications of information, but what it cannot see is anything at the gene level, so it doesn't look that closely. It can't see anything at the base level or at the gene level, and it also can't see any structural changes. So things like balanced rearrangements, which kids with PMS don't have, but some of the parents can have a balanced rearrangement or translocation, or other structural changes like ring chromosomes, a microarray won't be able to see. If we think of this test in the library, what it's doing is it's putting on binoculars and looking all the way in and saying, is this book here, is this book here, is this chapter here, is this chapter here? And it does this over and over and over and over, but it's not able to get a sense of the overall structure of the library or if the bookshelves are organized properly. The next test that is commonly ordered that can diagnose Phelan-McDermid syndrome is a karyotype. So this is the picture that we looked at on the first slide, which again takes a picture of our chromosomes and then lines them up. What a karyotype can see are larger deletions or duplications or other rearrangements. It can see things like balanced rearrangements and other structural changes like ring chromosomes, but it can't see deletions that are below a certain size. So there may be some people who have a microarray that shows a deletion, but then a karyotype that's completely normal, which can be confusing, but typically that's just because the deletion is too small to be seen on the karyotype. It also doesn't see at the gene level. It definitely doesn't look that closely, so it can't see sequence variants. If we think of this test being done at the library, what it's doing is looking overall at the bookshelves. Are they all here, and are they all in the correct place? Testing for mutations, again, there's different types of testing, but the underlying technology is the same in that it's all a different type of sequencing testing. So there's exome sequencing, genome sequencing, and panel testing. Panel testing is when they sequence just specific genes associated with a certain symptom. So there's autism panels, seizure panels, hypotonia panels, but again, what they're doing is the same of reading through each gene. What sequencing tests can see are sequence variants or mutations in SHANK3. And more recently, they've also been starting to report back deletions, so chromosomal deletions on an exome sequence. So people who have been diagnosed in the last few years may have had a deletion be reported back from an exome sequence. Sequencing testing cannot see balanced rearrangements, again, which some parents may have, or ring chromosomes or anything else structural. If we think of this at the library, what this test is doing is it's reading through every letter of every book to see if all sentences make sense, if all the stories make sense. So now is the time to break out your phones, take out your reports, and we will go over the structure and the format of genetics, and then we'll look at a few different examples to see that they can be reported differently, but the content is always the same. Again, we will start with deletions and start with microarrays and then move on to mutations. So depending on the year, depending on the lab, depending on a lot of things, all of your reports look a little bit different, but they all hold one sentence that will be in there at some point in your report. And this one line is really the bulk of the information that you should take away from the report. So it always starts off with the letters ARR, and that tells you what I'm about to tell you is the result of a microarray. After that is the reference build, so what they compared it to. For most people, it will say GRCH37 or HG19, which is the same. If your report is a little bit older, it might say NCBI36, and if your report's very new, it might say 38. This isn't so important for parents to understand, but as researchers, it's important for us because when we compare a group of kids who have PMS, we like to line up all the deletions on the same build. The next piece of this line will tell you the region of the chromosome that they found a difference or a change in. So they'll all say 22, the 22nd chromosome, Q, the long arm, and then 1,3 again, which is the band of the chromosome that is affected in PMS. Some reports and some labs give additional information, so it might say Q13.33 or Q13.31-33. Depending on the lab, some give a little more, some give a little less information. But overall, most people or everyone will say Q13. The next piece is the one that is specific to everyone's individual child. So these are the exact coordinates that are deleted or missing in your loved one. So the first coordinate is the start coordinate of the deletion, and then the second coordinate, or the second number, is the end coordinate of the deletion. So this is the range that they don't see the right amount of information in. If you subtract these numbers from each other, you'll also find the deletion size in base pairs. And then the last thing you'll see on the report is times 1. And this is telling you that it only saw this information one time, whereas because we all have 2 copies of chromosome 22, we should have seen it 2 times. So times 1 is indicating there's a deletion. For other genetic conditions that are caused by duplications, you would see a times 3 there, which again means you saw it 3 times, which is 1 more than is typical. So in your many pages of report, I know there's a lot of words and a lot of numbers, but this is truly the one line that you, I hope, can take away from it. Here are some examples of different types of microarray reports. So in this one, they give results and interpretation, where they talk about the deletion, they talk about the size, they mention Shank3, and then near the end of the page, they give you this sentence formatted. So again, they tell you it's an array, they tell you the build, they tell you the chromosome, 22q13.33, the start coordinate, the end coordinate, and then times 1. In another example, this lab chose to report it out as a table. So they start with CNV, which stands for copy number variant. Loss means a deletion. They give you chromosome 22, band Q13.33, the start coordinate, the end coordinate, and then they give you the size in kilobases, which is typically easier than giving base pairs. And then I think it was on the second page or near the bottom also of that report, you see the standardized one line of the results written out. So it may take some searching if labs prefer to give it as a table than as a line, but the majority of people should have this one line of information. And if you don't, feel free to come see me and we can read through it together. For sequencing testing, so the same thing applies here depending on the lab, depending on the year. It will all look a little bit different, but most labs standardize one specific table that they'll give you right up top. So this starts with gene, and for some people who did an exome or a very big panel, there is the possibility of seeing more than one variant reported here, but you can follow along just on the Shank3 line for now. So the gene they tell you is Shank3, which is the one that's affected. The disease will say Phelan-McDermid syndrome. It might also say Shank3 disorder or Shank3 related disorder or Shank3 related PMS, but those are all the same thing. Next is the mode of inheritance, and this is in reference to the disease or the disorder. We know that Phelan-McDermid syndrome is a dominant disorder, meaning of the 2 copies of Shank3 that we have, only one needs to be affected for somebody to have the disorder. This opposes to recessive conditions where both copies need a change for somebody to have the disorder. Those are things like cystic fibrosis or Tay-Sachs or other conditions you may have heard of. Next is the variant. So for people who have a child with a mutation, this is the box that tells you the specific information for your child. So it will always give you 2 different sets of letters and numbers. The first one starts with a C dot, and this tells you what happened to the DNA. So here it's telling you that at position 3679 in the Shank3 gene, the base that's there has been duplicated. The base that's there is a G, so for you or I, at that position we have a G. For someone who has this mutation, they have 2 Gs. The second line starts with P dot, and this tells you what happened to the protein. So this is telling you that there used to be one amino acid, alanine. It changed to a different amino acid, a glycine. And again, when we think back to how our body reads through genes, we know that if we duplicate just one base, it's going to affect that 3-letter sequence, but then also everything after it. That's called the frameshift variant. And so after the GLY, which stands for glycine, you'll see FS, which stands for frameshift. So this is telling you the type of variant that this is a frameshift mutation. And then the star 69 means that it tried to continue reading those 3 sequence pairs, but after 69 times, it read a stop, which is early in the gene. The specific variant here is not applicable to everyone because everyone has a different mutation. I chose this one, though, because it is the most common in PMS, so I think, at least at our site, we know of at least 30 or 40 kids that all have the same exact mutation. But there are certainly hundreds of other mutations that we see as well. Zygosity is asking, how many times did we see this variant? Heterozygous means that we saw it on one copy of SHANK3 and not the other. Again, because we know this is a dominant condition, we're only expecting to see it on one copy and not the other. Inherited from, so this will be reported back if parental samples were given for the sequencing test. De novo means that the variant is new in the child and wasn't inherited from the mom or the dad, which we should see on mostly all of these reports. If parental samples were not given, it might say not known or not applicable, and if only mom or only dad sample was given, it could say not maternal or not paternal. And then the last box that it will give you is classification. So the way that sequencing works is they read through your child's gene and they compare it to a reference. And every time there's a change, they're going to scale it or rate it on a scale from benign, meaning we know for certain that this is just a normal change between people, to pathogenic, meaning we know for certain that this change affects the meaning of the gene. To have a diagnosis of Phelan-McDermid syndrome, this box will always say pathogenic or likely pathogenic. I think I have, no. So I didn't put examples of sequencing testing because they all should have this one table in there. But again, if you're having trouble or you don't see it, come find me after and we can review together. For this, I don't want you to look too hard at it, but for people who are more visual learners, I just wanted to give a representation of what the SHANK3 gene looks like and then what somebody with this mutation, what their gene looks like. Again, certainly don't read through this. The only thing I wanted you to take away is just to see the longer sequence that you see is a full SHANK3, and then on the other side, you see the SHANK3 of someone who has this specific mutation. What you can see from this is the first red letter on the longer side you can see is an A, which stands for the alanine that you or I probably have there. And then it's changed on the shorter gene to a G, which stands for glycine. And you can see that after this A and this G change, everything after that has also been changed for this person who has this mutation. And again, you can see that the gene ended early after 69 kind of attempts of continuing to be read. Really, to take away is just the length of each gene to show that it has been shortened in someone who has Phelan-McDermid syndrome. Follow-up testing. So this will move on a little bit from Genetics 101 and reports. But if you have questions, there'll be time at the end for group questions, which you think would pertain to more than just your child. For any questions that are specific, I'll walk around and answer questions, or you guys can find me after, and we can talk about specific reports. For follow-up testing, so there is a set of recommendations that we have for people who are diagnosed with PMS of what to do next. This is also listed out on the PMSF website, so they give you this PDF which shows you based on if your child has a deletion or a mutation, what testing should you have, but I will also walk you through it now. So the last hard genetics concept I will throw at you is something called mosaicism. Mosaicism is when somebody can have a different genetic makeup in different cells of their body. So it's possible for someone to have a SHANK3 change in some of their cells, but not all of them. This person won't necessarily have symptoms of PMS or any sort of disorder, but for them, they have a likelihood of having one child with PMS, but also having a second child with PMS. The reason that we look for mosaicism is to help with family planning, for people to understand if you already have one child with PMS, do you have any increased likelihood compared to the general population of having a second child with PMS? Some of the follow-up testing is related to mosaicism, which is why I'm explaining it. So back to general follow-up testing for people who have a child with deletions. I started this with people who have a microarray since that's the most common. So if you've had a microarray, what to do next? Again, we know that microarrays look at the amount of genetic information that we have, but not at anything structural. So the first thing that we want to do is recommend a karyotype, which can look for a ring chromosome. The reason we recommend that is because there's additional medical recommendations for kids who have a ring chromosome compared to kids with just a deletion. So it's useful for you as parents to know if you should be following these extra recommendations or not. If your child does have a ring chromosome 22, we recommend a karyotype for a parent to look for a mosaic ring 22. Again, not looking to diagnose PMS in a parent, but really hoping to help with family planning if you're thinking of having more kids, to have an understanding of if there's an increased likelihood of having a second child with PMS. A karyotype can also look for balanced translocations, but I included that on the second slide to split them up a little bit. FISH testing is also recommended. We haven't spoken about that yet, but what FISH testing does is it assigns colors or highlighters to different regions of different chromosomes so that you can look at it and visualize the organization of our chromosomes. What this can do, and what karyotypes can also do, is look for balanced translocations. So this in a parent, we know that people with a balanced translocation have the right amount of genetic information, but the chromosomes are stored differently. So here in the example, you can see that some of the orange chromosome and some of the green chromosome have swapped information. This individual won't have any sort of disorder, but when they have children, there's a possibility that they will pass on an unbalanced version of this. And for these individuals, they also have an increased likelihood of having a second child with PMS, which is why we recommend to have this testing done. The other thing about balanced translocations is even if you yourself are not thinking about having additional children, typically balanced translocations we see kind of passed down from parents to kids to kids to kids. And so if you carry a balanced translocation, it's likely not new in you. It likely came from one of your parents, and your siblings might also have one. So again, if you're done having kids but you have siblings, it can also be useful just for family planning for everyone in the family. The last test that we recommend for individuals with a deletion is a urine screen. This looks for a condition that's associated with a gene called ARSA, which is right before SHANK3. ARSA is associated with a recessive condition, meaning that both copies need to have a change for someone to have the disorder. For people who have a deletion, typically it also includes ARSA because it's so close to SHANK3. But we have the other copy, or your child has the other copy that's working completely fine. In very, very rare cases, that other copy of ARSA can have a mutation, and that person would have another genetic disorder. Again, it's rare, but because the screening can be done on a urine screen and it's pretty simple, and there would be additional medical recommendations, we do recommend for you to ask your doctors to have this screen. This is all of the recommendations for people who have deletions. If you didn't start with a microarray and you started with a karyotype, for example, we would just recommend the other three. So there's four tests overall. There's the microarray, a karyotype, FISH testing, and the urine screen that people with deletions we recommend to have. For mutations, the list is a little shorter. Help. There, okay. Mutations, the only one that we recommend is just for de novo testing, which is testing of the parents. So if both parents are available, we recommend to send samples in to look for the sequence change to show that it's de novo, or in rare cases to show that a parent might have mosaicism. And this again is for family planning to understand if you want more children, what the chances are of having a second child with PMS. That is all the information. So I will take general questions now, and then I'll also walk around the room and talk about individual questions. The other thing is that the PMSF offers genetic counseling with me. So I meet with one family a week. It can either be newly diagnosed, or if your child was diagnosed 30 years ago but still have genetic questions, I will see anyone. The only thing to do is just email Carla with your genetic report, and then she'll connect us and we'll set up a time to talk. We'll have 45 minutes to kind of talk about genetics or PMS or research or really anything that you'd like. So that's this program. And then here's our very wonderful team of people at the Seaver Center and the RDCRN, the PMSF, and then all of our PMS families that we cherish.
Thank you so much, Tess. I think I speak for everyone. That was incredibly clear and really, really helpful. I think we will do about 10 minutes of general questions, and then as Tess said, she can do some more hands-on or individual questions throughout the conference. I'm sure she'll be inundated because she's so helpful, but if you see her, you can talk through more individual questions. So scan the QR code that's up here and enter in your own questions and or vote for questions that you also want answered that are more general. And I'll just give it a minute for things to populate, and then I'll start asking. And we'll end a little bit early so that Tess can talk to folks one-on-one. Okay, I love this question. What is the difference between whole exome sequencing and whole genome sequencing?
Great question. So an exome sequence looks at the part of genes that directly go into making a protein. There's a lot of genetic material in between genes and in between parts of genes that don't directly go to making the protein. The exome is definitely thought to be the most important part of the genome, but it also only makes up about 2 to 3% of the genome. Whole exome sequencing right now is the best and most comprehensive clinical test. Whole genome sequencing is starting to be ordered as a clinical test, but oftentimes we're still only interpreting the exome. So right now there's not a huge difference in terms of what you would receive back from testing, but genome would cover kind of additional genetic material.
I think a common question is, as a follow-up to that, if someone got whole exome and they got a positive result, so they're diagnosed with Phelan-McDermid, they don't necessarily need to get whole genome or anything like that. So just because there's different testing doesn't mean they have, as long as you have that result from sequencing. Is that correct?
Yeah, I think this answer will differ depending on who you ask. When families ask me if they should pursue additional testing, for example, if they have a microarray that showed a deletion, should they have whole exome sequencing to look at all the other genes? Typically, I answer that if you think that the features of PMS fit with your child well, then personally, I don't see a point of continuing to search for more, just because the more you look, kind of the more you find and the more uncertainty you find. But for people who have children who they think some of the features overlap with PMS, but their children have these things that don't fit with PMS, they don't see it reported, for those individuals, I would recommend to pursue other testing or see what their individual doctors recommend for them.
Thank you. I think we have a couple of great top questions here. The first one is, do siblings need to be tested for their family planning?
So the only cases where siblings we would recommend to be testing is if there's a balanced translocation that's in the family. Otherwise, siblings don't have any additional risk of having or any additional probability of having a child with PMS compared to the general population.
If there is a balanced translocation, is what you said. Okay.
Yeah.
Does the size of deletion indicate the severity of the condition?
Yes and no. I could talk for like 5 hours about this, but I will condense it into like 2 sentences. In some cases, yes. So there's some findings that the larger the deletion, there's some specific findings that we only see in kids who have larger deletions. These are things like kidney problems and lymphedema, where kids with very small deletions or mutations, we never see them at all. For these findings, we have a good understanding that it's likely not CHANK3 that's causing them and genes that are further on in the chromosome. The other kind of consistent findings that we see are kids that have larger deletions tend to have more severe developmental delays. So they're walking later, they're talking later, the likelihood that they'll speak verbally is lower than kids who have very small deletions or mutations. At the same time, we see higher reports of regression in kids who have the smaller deletions and the mutations, but we're trying to tease apart if there's some sort of genetic risk that's playing into this higher risk of regression, or if it's because these kids have more skills to begin with, and therefore we're better able to measure that skill loss or regression. So yes and no. There's also a lot of, bias in looking at genotype-phenotype studies where we compare the different genetics to the different clinical features. A lot of this is just in terms of who is diagnosed and what insurance carriers take as an indication for genetic testing. So there's still quite a bit of uncertainty, and it's certainly not like a one-to-one of this is my child's deletion size, can I predict how they will be in the future? But those are the kind of consistent findings across cohorts are the ones that I just mentioned.
Can you give general ranges for sizes of deletions?
So sizes of deletion range from very small of just portions of SHANK3 that are deleted. So this would be like maybe 50 kilobases is around the smallest, and then the largest that we see can be about 9 megabases. So for numbers of scale, you can think of from under 1 to over 9 is the scale that we typically see.
Is there a need to repeat testing as the years progress and technology improves? Say we had testing done 7 years ago.
No. I don't personally think so. I mean, the technology changes, but somebody's genetics don't change. So if a deletion was found 10 years ago and you look again today, the same deletion will be found. Perhaps the specific coordinates might be a little bit more accurate today than 10 years ago, but the general idea is, the general finding is the same.
Can I ask a follow-up on that for if it's a variant in SHANK3, if it's likely pathogenic when they get it or it's unknown significance, is it important for them to have it reinterpreted later as more data is collected or no?
Yes, so likely pathogenic is accepted as a diagnosis. Oftentimes it's reported as likely pathogenic and not pathogenic because there's just not enough points to get it to pathogenic. Maybe if there wasn't parental samples available or some other information, but if it's likely pathogenic, you can be confident that that's a diagnosis. For anything that's reported as uncertain, technically it's not a diagnosis of Phelan-McDermid syndrome or anything else in a gene that's reported as uncertain. But yes, it's a good idea if you have an uncertain variant to check in with the lab every year or so just to see if they gained any new information that could help reclassify it either down to benign or up to pathogenic.
It's interesting because I guess those folks wouldn't necessarily be with us because they wouldn't have received a diagnosis, but
Yeah.
that makes sense. If ring 22 always involves a deletion on the end of the gene, why doesn't this get reported on the microarray?
Yeah, it's a good question. It just depends on how many markers are in each part of the microarray, and typically they report back things that have clinical significance. On the short arm of chromosome 22, there's not much going on there, and so they're not going to report back a finding that doesn't have clinical significance for you.
What are some of the things for ring 22 that you mentioned, or are there just generally, and then I can mention a couple places where people can find more information?
Yeah, there is a lot of information, definitely in the medical recommendations that were just published, of just some additional recommendations for people with a ring 22. Some of this centers over additional MRIs that are recommended, or hearing screening, or I think there's also some dermatology screening. But I'm happy to walk through the recommendations with anyone to show you what additional would be recommended.
And that might be covered today. And I also see Dr. Katie Phelan here, who's an expert in it, and I know that she's published a lot on ring 22. So, and Dr. Curtis Rogers is also in the audience. So there are a lot of resources. I think there are pages that have been published on NORD and elsewhere, but if anybody wants to add anything there, we can give you the mic. But yes, it's connected with neurofibromatosis type 2, which can cause, I think, non-cancerous tumors in the CNS, so they just need to be monitored. But please add anything if you'd like. So I think we'll maybe take one or two more questions, and then we'll end so that folks can ask Tess individually. Let me see. Do you ever see gains or additions in addition to deletions, or both?
Yeah, in terms of like a duplication of SHANK3, I know that there are some, but it's not going to cause the same symptoms of Phelan-McDermid syndrome that we see when there's a deletion of SHANK3.
And the last question is, what is the significance of, you mentioned you have the protein information. What is the significance of that? Does it determine additional symptoms based on, what the protein is or what the change to the protein is?
Yeah, it's a good question. So with the mutations, what the current theory or the current hypothesis is that no matter what the sequence change is, it's causing the gene to end early, and we're just not making SHANK3 protein from that copy of the gene. So whether your change is at position 1 or 1,000 or 2,000, it's just causing that copy not to be functional. So the thought right now is no, it doesn't actually matter where the change is and what the specific change is, but it will result in premature termination of the gene. We'll see if that changes as research evolves, but that's the current thinking.
Thank you so much, Tess. And I think there will probably be a lot of individual questions. We really appreciate it.